now the remaining part of the digestive system. So far you have learnt about the structure of digestive tract, and under digestive glands you have learnt about the salivary glands, the gastric glands, then intestinal glands, then liver. Part of the liver we have already completed here now. We have talked about some of the important functions of liver. Then the remaining functions of the liver we shall talk about now. One of the most important functions of liver is phagocytic in function here now. Liver is highly phagocytic. Within the liver there are certain cells called Kupffer cells. Kupffer cells. These Kupffer cells, with the help of Kupffer cells here now, it renders phagocytic activity. With the help of Kupffer cells, it renders phagocytic activity. Dear students, you remember this point. This is the most important point for all the competitive exams, AIMS, JIPMER, AFMC, CMC and all that. There were several times, many times they asked the question based on this Kupffer cells. So remember, Kupffer cells are the phagocytic cells in the liver. Okay. Then another important function of liver is it synthesizes urea. Liver synthesizes urea through ornithine cycle. Synthesizes urea through ornithine cycle. This ornithine cycle is also called Krebs-Hanselit cycle. It is also called Krebs-Hanselit cycle. Ornithine cycle is also called Krebs-Hanselit cycle, or it is also called urea cycle. Now, why is it called ornithine cycle? It involves one of the intermediate products called ornithine. Ornithine, citrulline, arginine. Ornithine, citrulline, arginine are the intermediate products of ornithine cycle. This we shall take up it later on in detail here now. Later on I shall give you the entire cyclic representation of the ornithine cycle. But time being, you remember, ornithine cycle is also called urea cycle or Krebs-Hanselit cycle. It takes place in liver and it is associated with the formation of urea. The intermediate products of this cycle are ornithine, citrulline and arginine. In the presence of an enzyme called arginase, formation of urea takes place. Even this question, many times it was asked in various competitive exams. They asked the question like formation of urea in human being takes place in number 1 liver, number 2 kidney, number 3 digestive tract, number 4 say lungs. So you should definitely answer liver. When both the options are given, liver and kidney, most of the students are tempted to answer kidney because kidney is the main organ of excretion here now. So they are tempted to answer kidney, but kidney is not the answer here now. Kidneys are meant for only elimination of urea, but not synthesis of urea. Formation of urea takes place in liver through ornithine cycle here now. Then how is urea synthesized? Urea is synthesized through a process called deamination. What is deamination? Removal of amine group from an amino acid here. I'll show you the empirical formula of an amino acid here now. RCHNH2. Okay, RCHNH2. This carbon on one side has an alkyl radical group which varies from amino acid to amino acid. On the other side it has amine group, and on one more side it has a carboxylic group here now. It has a carboxylic group. So during the formation of urea here now, this amine group is removed from the amino acid. It combines with carbon dioxide to form CONH2 taken twice. Combines with the CO2 to form CONH2 taken twice, which is otherwise called urea. So since formation of urea occurs by removal of amine group from an amino acid, it is called deamination process. So students remember, liver is also a site of deamination process in the human body here now. Apart from this, liver also facilitates transamination process. What is transamination? Conversion of amino acid from one form to another form is called transamination. Like there are two kinds of amino acids, essential amino acids and non-essential amino acids. The non-essential amino acids are synthesized within the body. Essential amino acids are taken along with the foodstuffs. Sometimes there will be transformation of amino acid from one form to another form, maybe essential to non-essential and vice versa. So that also takes place in liver, which is referred to as transamination process. So it also occurs in liver. Then liver also synthesizes albumin and globulins. That is, liver also synthesizes albumin, globulin and heparin. It is a mainstay for the synthesis of heparin. Remember, this is the most important point once again. Bear in your mind for AFMC, JIPMA, CMC, so on and so forth here now. In human body, formation of heparin takes place, number 1 in liver, number 2 basophils, number 3 mast cells. So they are the three sources from where heparin is synthesized: mast cells, basophils and liver. What is the significance of heparin? It acts as intravascular anticoagulant. It acts as intravascular anticoagulant. Albumin helps in maintaining the viscosity of the blood. It is a protein, helps in maintaining viscosity of the blood, and thereby it helps in retention of water in the blood. Globulins give rise to antibodies. Globulins are the proteins which give rise to antibodies. So apart from blood coagulating factors like fibrinogen, prothrombin, then factor number 7, then 9 and 10, even albumin, globulin and heparin are also synthesized in liver. You get a question in the MSET or any other exam. What are the units of liver? What will you answer here now? You have to answer liver lobule. Liver lobule is the unit of liver. Unit of liver. Functional unit of liver is liver lobule. This also you have learnt already. Liver has two lobes, right lobe and left lobe. Right lobe is once again divided into right lobe proper, caudate lobe and quadrate lobe. Within each lobe there are lobules. So lobule is referred to as the functional unit. Liver lobule is referred to as functional unit of liver. And each liver lobule is covered by a connective tissue sheath. Covered by a connective tissue sheath called Glissen's capsule. Covered by a connective tissue sheath called Glissen's capsule. So that is also most important point. I have already made a mention about that. Glissen's capsule is a connective tissue sheath that covers each liver lobule.